Either way, it's one.
Yeah, that's
That's the one I really am paying attention to because I do my own. Everybody jumping. Yes, but the compulsory do it. So it's I know that's like when people like, yeah, I like how they're doing everything like we're not doing like but it's like the code.
so I say yeah because my
I know, I guess.
like you know what?
I don't have to do it because I do anyway. I know, right? When I saw the date, I don't care.
I feel like that's how I
Yeah, by the time you get back,
I don't know.
I'm trying to think of like one thing that I usually like right now where it's like I'm just tired of being like tired of so good. We're like reality is like yes it's compulsory but it's actually like not macro and then it's like all the new people and I'm like I still don't know. And it's just like it's pure like it's just like aggressive.
What? We already turned it in, and I got it ready.
What brand is it?
So smart of you. Absolutely. It's like a small shot.
like the great part versus like the cutoff.
I'll cut it out and put it on.
I think for sports.
I'm gonna tell you, Lane, to tell her dad. He works for the school. What does her dad do?
Yeah.
He's like the dean or something. Lane's dad.
I forgot what he does, but I had a. No, I don't even know. Like literally,
I'm not doing it next year.
like it doesn't.
Remember, you always say this.
Yeah, no.
Always say this. I'm serious.
Like putting their names like Coach Brady.
I'm gonna do it in that credit front,
like I said, like my name. So like. Like just put it out.
You know what?
That makes sense. Like it literally just.
Next year, we're taking I'm taking who I want. I'm taking it. Not gonna do it. You have to learn it back.
Yeah.
It's got a stem, dude.
All right, guys. Good morning.
Good morning.
Hello.
Hello.
Happy Valentine's Day. So did you guys get a chance to look at your exams and the answer key and get a second to look at what, questions you got wrong? With that, were you guys able to understand why you got the question wrong? Was it clear after going through? Okay. Yeah.
Are we allowed to ask questions?
Yes, you are allowed to ask questions. It was just for 24 hours.
The question that I had, it was about the temperature changing, and I was just gonna ask why the flavonoids was not the answer, and that the lipids was the answer.
Yeah, so that question only talked about temperature changing, which means it could go up or down, which means hopanoids are usually stiffening agents, so they usually are made when you want to stiffen the membrane, when there's an increase in temperature. but the question asked about just any change in temperature, and so usually with any change in temperature, there's going to be some modification of the lipid structure. That could include hopanoids, it could not. It's just a more general answer. Hopanoids are more specific to just having an increase in temperature.
And that would go for the same for the cyclic question?
Yes.
Like for A.
Yes,
Okay.
same thing, yeah.
Thank you.
Yep. Any other questions? Okay, so, bless you. Just a reminder, worksheet 3 is due on Monday. It's already up on Blackboard. And your second exam is in 2 weeks from today, so the last day of February. So it's gonna creep up on us. It will be on, less amount of material than first exam because it's a shorter amount of time. But please make sure that, we're done with about half of the material for this exam after today. So please make sure that you come to class on Monday. That worksheet's going to be really helpful in just making sure you're all caught up on all this information. Remember, we talked about viruses and phage, and then we switched directly into bacterial transcription and translation. Today we're going to talk about what happens after translation and after we make proteins. So make sure you take this time this weekend to study, and in 2 weeks we'll have our second exam. So it will creep up, but I think it might be nice so that you guys can go off on spring break and not have to study for this class, so we can kind of start fresh right after spring break. So yeah, any questions on that? Okay, awesome. So today we're going to talk about, we're going to go through translation again, going from mRNA to protein, just adding in some more details to make sure that you guys really understand it. And then we're going to talk about, what happens to the proteins after they become translated. They can be modified, folded, or degraded. And so also we'll talk about some protein traffic control. So how they are exported essentially. I should probably start with taking attendance. All right, Riley? I'm gonna have the classes missing today. Brianna? Drew? Henry?
Here.
Nick? Abby? Hannah?
Here.
Rosa?
Here.
Matt? Jillian?
Here.
Mia?
Here.
Annie? Brianna? Sadie?
Here.
Jessica?
Here.
No, don't call Jessica. I said Sadie and then Jessica, and I heard Siri call Jessica. Jesse?
Here.
William? Marissa? Anna? Aubrey? Matthias? Colin?
Here.
Olivia? Amber?
Here.
Katie?
Here.
Kennedy? Ben? Ava? Kendall?
Here.
Emily?
Here.
Lauren?
Here.
Jennifer?
Here.
Isabel?
Here.
And Corwin. Okay. Give me 2 seconds. I'm just gonna answer it up.
Did you see them? I did. And then the ones that she originally said, like, oh, there's some things from. I didn't even see that those posted that. That's so funny. She said, well, like, I look cute. I know, exactly. I'm like.
any picture we take of our
Okay. So let's go back a little bit and just, it's helpful to look at what we've learned so far about, taking a DNA template, using an RNA polymerase in order to make the mRNA transcript, and then the ribosome can then make a translated protein. So at the top, you have that sense strand of DNA. So up here, sense strand just means that it's the one that can get coded because it's in the 5' to 3' end. And then you have the bottom, which is the template strand. You also have, this is DNA with 2 genes on it. We call it a 2-gene operon. So you have gene A and gene B that are sitting on this DNA strand. And so the template strand is what's read by the mRNA because it's in 3' to 5', and the newly synthesized strand is going to be synthesized from the 5' to the 3' end. That's why it's called the coding strand. So this is the direction of transcription. And so remember, there are elements on the DNA, which is the promoter. So you have this promoter right here, and can anybody remind me what it is that brings the RNA polymerase to the promoter to do transcription? Yeah. Sigma factor, yep. So sigma factor brings the RNA polymerase to the promoter site so that transcription can then start. And so that eventually will result in an mRNA transcript where you have 2 start codons, one here and one here, and 2 stop codons. So can anybody tell me why we have 2?
In case one messes up.
Possibly. There's a more direct answer just based on what you see here. Yeah, what were you gonna say? Yeah, there's 2 genes, so you need a promoter for each one. So you have this, and so you can essentially have one promoter that will be for both genes. That's if they're like right next to each other and they have similar functions, but sometimes you'll have more than one as well. So you essentially then make the mRNA, and then the mRNA is then translated into these 2 proteins. There's 2 because there was 2 genes to start with. Does that make sense? Okay, so we'll kind of then fast forward a little bit back to the ribosome and the mechanism of translation. First, the ribosome has to get properly positioned on the mRNA transcript so that peptide synthesis can begin. In initiation, remember there are 2 ribosomal subunits, the 50S and the 30S, that have to then be brought together. And so then when they're brought together to make that whole ribosome, then it's placed on the first amino acid position, which is usually the start codon. Does anybody remember what the start codon is? AUG. Yep. And then what's the amino acid that codes for that codon? Methionine, yeah. Good job. So, be careful when it comes to the wording. If I ask for the codon, it's AUG. If I ask for the amino acid for the start codon, it's methionine. And so then you have elongation, which is the sequential addition of the amino acids, and that's directed by the mRNA transcript. And then termination is when you can release the completed protein, and then those ribosomal subunits get recycled for use for more protein synthesis later. Each one of these phases requires these protein factors and energy. That energy is in the form of GTP. So remember, the first amino acid is the start codon amino acid, which is methionine. And so that usually, remember, comes from the tRNA. The transfer RNA has to bring that to the ribosome. And it has to be charged. So when it becomes charged and the tRNA is able to actually bind it, it becomes N-formal methionine. So that's essentially, the methionine, but it's charged. And so that attached to the tRNA then allows the ribosome to recognize it. And so basically this is the only tRNA amino acid combination that can bind directly to the P site. P site, remember, there is what are the three sites that we talked about in a ribosome?
A site, B site, B site.
Yeah, A site, P site, E site. So that N-formal methionine, or that methionine, is the first amino acid that comes along with the tRNA, and it can bind directly to that P site. And so the elongation step, these are essentially the three steps that you guys should know. They're repeated over and over and over again. So you have that charged tRNA bound to the acceptor site. Then you have a peptide bond that forms between the new amino acid and the growing peptide that's pre-positioned in the P site. And then the message will then shift by one codon in order to have that uncharged tRNA eventually move to the E site, which is the exit site, and then it's ejected, and then it allows for another tRNA to come in through the A site that's charged. So this is a cycle. It happens over and over again. And then in termination, you release the completed protein, and you can recycle those ribosomal subunits. Can anybody tell me what the ribosome has to arrive at for it to then terminate and know that there's completion of translation? Yeah. That was transcription. Yeah, see, it's easy to confuse the two.
I feel like I know it.
I'll give you a chance. No? Yeah. The stop codon, remember? See, those are the things that you guys, it's easy to confuse between transcription and translation. You have, the hairpin loop, which is the row-independent termination, and then you have the row-dependent termination, which is that protein for transcription. But this is translation. Translation, we have codons. And so you have start codon, and then you have 3 stop codons that can signal the end of translation. Does that make sense? So I do personally like emphasizing how antibiotics work, and especially antibiotics that can inhibit protein synthesis in bacteria. And so given this importance of protein synthesis, and given that the ribosome in bacteria is very different than the ribosome in eukaryotes, in our cells, It's actually an excellent target for antibiotics because it's used for this fundamental process that bacteria need to survive with little chance of cross-talking with eukaryotic ribosomes, with our ribosomes. So I want you guys to, I'm going to talk about 4 antibiotics that inhibit protein synthesis in bacteria, and I don't want you guys to know the structures, I just want you to know the mechanisms. So streptomycin is an antibiotic that will bind to the A site. So when it binds to the A site, then it allows for improper codon-anticodon matchups. So it's essentially binding where the tRNA is supposed to bind. And so you have an improper, wrong protein sequence that results. So translation still can occur, but then it will kill the cell because it's made the wrong protein. So although translation can still occur in this case, it's just being mistranslated because now you have something else binding to the A site. It's going to inhibit the proper protein synthesis, and that will then kill the cell because it's not making the protein it needs. Any questions on that? The second one they need to know is tetracycline. Instead of binding to the A site, it actually binds near the A site. So it kind of blocks and prevents that charged tRNA, which is the aminoacyl tRNA. When it's charged, it has that aminoacyl group attached. It prevents that charged tRNA from binding to the A site entirely. So if it's going to inhibit tRNA binding, then it's going to not allow for any translation to happen. So it acts as the steric hindrance by blocking the A site so the aminoacyl tRNA can't bind. So over here, they say that it binds to the A site, it actually binds near the A site. Make sure you guys write that in your notes. So that's why it's important for you guys to understand what's happening at the 3 sites of the ribosome, the A, P, and E sites, because these are targets that we can use for antibiotics, and they all work differently. Okay, chloramphenicol. This is one that we actually I will make plates basically that have this antibiotic, chloramphenicol, on it. Basically, this will then bind to the P site, and by doing so, it will inhibit peptide bond formation. So can the charged tRNA, can it still get into the ribosome, or can it still bind in the ribosome or no?
Yes.
Yep, it can because it's binding to the P site. So the tRNA can still get into the ribosome, but This antibiotic binding to the P site actually prevents the enzymatic activity of the ribosome because it's unable to make those peptide bonds to connect all the amino acids one after the other. And then the last one is erythromycin. And erythromycin also binds to the P site, just like chloramphenicol. And it also will alter peptide bond formation. So if you notice, it has this massive ring structure of carbon atoms, just like so many different carbon atoms. Like look at this huge structure. If you guys have taken some chemistry, you should, be a little bit familiar of structures like this. It means there's so many carbon-hydrogen bonds. And so by binding to the P site, it's altering, that peptide bond formation. So it screws with the amino acid sequence that will then come out of the ribosome. So the difference between these two is that this one will inhibit peptide bond formation. This one will alter the peptide bond formation. But they both bind to the P site. So these are all slightly different mechanisms and different targets within the ribosome, but ultimately they destroy protein synthesis and they destroy the cell. You should know where each one of these binds, similarities and differences between the two. So I could ask which two antibiotics will bind the P site. You should know this. I could ask, in the context of which antibiotics inhibit peptide bond formation, then you would know it's this because inhibit is the key word there. So make sure you keep, you really understand the differences there. Any questions? So we talked about very briefly other antibiotics in previous lectures for exam 1. Can anybody remember what those antibiotics targeted? These ones target protein synthesis. What did the others target? Something in bacteria that bacteria have, but we don't have. Yeah, cell wall synthesis, peptidoglycan. So there's different antibiotics that can target different processes in bacteria, and that's really important to know. But now we're talking about ones that inhibit protein synthesis. Okay. So finally, we've made a polypeptide sequence, but this isn't the end. The cell has to then make this polypeptide into a functional protein or an enzyme, so a protein that has enzymatic activity. So there's got to be some modification. So we tend to say that after translation we make a protein, but it's actually not entirely correct or accurate to say that. After translation we make the polypeptide sequence, but then there's got to be some protein modification, folding, and even potentially degradation that will allow it to become a functional protein. So for many proteins, translation is not the last step in producing this functional molecule. And so in order for it to actually have some sort of activity, it's got to be modified and it's got to have some 3D structure for it to be able to have that type of activity. There are different structures that eventually are made. So you have the primary structure, secondary structure, tertiary structure of proteins, just different levels of making it into this 3D molecule. If the protein is damaged, then the cell also will have mechanisms to remove it from the cell. So it'll clean the house essentially by degrading damaged or unneeded proteins. So after translation, one of the processing steps is that a lot of these proteins get cleaved to basically activate or inactivate the protein. What a lot of proteins and bacteria do is they have that first amino acid, N-formyl methionine, they have that removed. This only happens in bacteria, not in eukaryotes, so you can really take advantage of this for human health. So this is over here a cool, essentially a pretty cool video here where you have the bacteria and then you have a neutrophil. A neutrophil is an immune cell that will help protect from infection. And so if you have a bacteria, neutrophils will start chasing the bacteria to try to, remove the bacteria or kill the bacteria from the cell. So this neutrophil can actually find that bacteria by sensing the release of the methionine that's being released from the bacteria. So it detects that N-formyl methionine that's made by the bacteria. So we'll look at this. Let me see. That's the neutrophil. That's a red blood cell, and that's the bacteria. And it's literally chasing the bacteria around because it's detecting the N-formyl methionine. Because one of those steps in protein processing is that cleavage of that first amino acid, that N-formyl methionine. So this is actually a really nice example of something called chemotaxis, which is essentially, chemo comes from the word of chemical. Taxis is, chasing something that's being released. So N-formyl methionine is being released from the bacteria. The neutrophil can then sense it in that extracellular environment and continually chase that, attract it. And so this is just essentially another video. The way to know that what it's chasing is N-formyl methionine is because if you take a pipette and you release N-formyl methionine, this is a neutrophil, you can see how it is basically specifically targeting the N-formyl methionine, because you don't know that in this image. I'm just telling you that. But there's a way to actually test that. And so this is a way that our immune system can just naturally target these foreign invaders by specifically targeting things that bacteria make and release that are not made by our own cells. So here's just a pipette that's being inserted into a cell and releasing N-formyl methionine just to show you that that's what the neutrophil is attracted to.
In this experiment, tiny amounts of chemoattractant are released from a micropipette. When neutrophils sense these compounds, they polarize and move towards the source.
Okay. Any questions on that? So there are a number of other post-translational modifications. So remember, I'm using different words to describe this. I said that there's protein processing after translation. Another word to describe this, or another phrase, is post-translational modifications. These are some other 6 ways in addition to just cleaving that amino acid, the N-formyl methionine. Generally speaking, this helps modulate the activity of these proteins and enzymes. They turn them on or off. There's methylation, phosphorylation, basically adding a phosphate group, adding a methyl group, adding a lipid, adding other groups. So you don't have to know the details of these, but you should know, the names of these. You should be familiar with all of these. So this is all at the very primary sequence level. So this is when we've made the polypeptide and, you know, we've modified it to then make this primary sequence of what the protein is. Now let's talk about folding that protein into a 3D structure so that it can have some sort of enzymatic activity. Some proteins can fold themselves on their own, but many require some help by chaperones. So chaperones are proteins that help fold other proteins. There are so many chaperones, but you'll only need to know these two systems because they actually make up the majority of the chaperones across bacteria. DNA-K is the first chaperone, and you can kind of think of it as like the palm of your hand. You have this unfolded polypeptide that's gone through modification, post-translational modifications, but it's unfolded, and essentially that polypeptide will land in your hand. There's some electrostatic interactions that essentially will lead to this biophysical cupping of that protein, which will then help it kind of clamp down and fold it. So we have the DNA-K chaperone here in green. In yellow, you have the protein. It kind of lands in the palm of that hand. And then there's electrostatic interactions, which we're not going to go into the details about, that will then clasp and kind of hold it and make sure that it gets properly folded. Protein that's helping another protein get folded. Does that make sense? You should know what a chaperone is, and this example of it, and just the general way that it works. It clamps down on the polypeptide to help it get folded. The second chaperones that you should know is this complex. Remember, a lot of times proteins also have other proteins that work together in this complex. So this is the GRO-EL and GRO-ES chaperone complex. This works in a very different way from the DNA-K. Basically, there is this complicated barrel, and so the barrel is essentially the GRO-EL. And then you have this GRO-ES that is essentially the cap to that big barrel. So when you have this GRO-ES controls what can go into and out of the GRO-EL barrel. So that GRO-ES cap, so you guys should kind of know these terms, that one is the barrel, one is the cap. So the GRO-ES cap in red will control the entrance and exit of that polypeptide sequence into that barrel chamber. And so within this barrel is where the protein is folded. ATP is used by both, but the ATP is not necessarily used to fold the protein. It just helps with the electrostatics of interacting with the chaperones, so what's eventually going to fold the protein is those electrostatic interactions. But ATP is controlling the entrance and the release of the protein from the chaperone. So ATP is necessary for the entrance of the polypeptide, and it allows capping by the GRO-ES. And then you have the folded protein, and the protein comes out, the cap is opened, and then that allows for another unfolded protein to come in, and then the cap closes up on it. Those electrostatic interactions causes it to fold. ATP allows the taking off of the cap, and then that protein is released. A new one can come in. So we'll watch a little video that kind of describes this. So we're zooming into a bacterial cell. So here we have the cap on the bottom. It doesn't really matter. This is the big barrel chamber, grow EL. This is the protein that needs to be folded. ATP comes in and allows for the cap to attach. Some facts. And then it can come out on the other end. And then there's a cap on this end as well. You don't have to know much about the charges, but just know that there are some electrostatic interactions that occur when the grow ES cap attaches to the grow EL chamber, and that allows the protein to be folded inside. So you should understand that and the role of ATP in this. Yeah.
Is it ATP just for the grow EL and ES or for the DNA?
It's just for the grow EL and ES. Or sorry, they both use ATP. Yeah, they both do. Yeah, sorry about that. But they don't help fold the protein, they just help to release it from the chaperone or allow, in this case, the grow ES to cap the chamber, the grow EL. Okay. Any questions on that? Okay. So what about if there's a protein that is, not necessarily good for the cell, it can hurt the cell. The cytoplasm has to constantly surveil for useless proteins or proteins that can hurt the cell and then therefore must be destroyed and then recycled. So those signals that allow for, the cytoplasm to be able to tell that there's a protein that's not, it needs to be degraded, there's signals on those proteins called degrons. Those degrons signal or dictate the stability of the proteins. There is an N-terminal rule that suggests that the amino acid on that N-terminal end of a protein correlates with its stability. For example, if there's leucine is one of the amino acids. It's very unstable. So that's one example of a degron signal. You don't have to know those details for this exam, but it's just, there's certain amino acids that are very unstable, and if they fall on the N-terminal end of the protein, that can signal this instability, and so that's a degron, essentially. And so when there is a degradation signal that's recognized, then the protein that's misfolded, for instance, can then be recognized by proteases. Proteases are proteins that have enzymatic activity to break down proteins. So proteases essentially will recognize the hydrophobic regions, but you don't need to know too much about that. But bacteria there's some examples of proteases, and so they're also ATP dependent, and so those are called endoproteases because they are inside of the cytoplasm. LON and CLP are some examples. I will show you a little bit about what a CLP looks like. But you should know what a protease is. You should know that a proteasome is the entire machine or complex of protein degradation. Proteosomes are found usually in eukaryotes and archaea, but in bacteria we have proteases. Those are dependent on ATP. But just so you know, we keep this in the context of just all forms of life. This bacterial CLP protease, so this is essentially a protein that breaks down misfolded proteins. The CLP in bacteria looks similar to something that would be found in a eukaryote or an archaea. So proteases and proteasomes recognize and degrade proteins. Also notice that the CLP protease, the structure of it is pretty similar. Oh, sorry, go ahead.
Is that the kind of microbe that?
Yeah, exactly. So when we usually will say a protease, it's, you know, it's essentially termed as just this whole thing is the protease. So it's for bacteria. A proteasome, because archaea and eukaryotes have just different processes for transcription and translation. There's usually, this is, you know, kind of split into multiple different parts that make up the proteasome. So that refers to eukaryotes and archaea. Yeah. But notice that the structure of this is similar to that GRO-EL, GRO-ES, where you have that inner chamber. The active sites are inside of this inner chamber in purple. And so this is where the protein comes in, and then you have these two caps on either side. And so instead of the protein coming in and getting folded, this time it gets degraded. These are ATP dependent, just like the chaperones. So when the protein finally exits the ribosome, the cell then has a choice. Do I fold it or do I destroy it? And this is really essential for bacterial survival mechanisms, especially during changes in environmental stressors. So damaged proteins will randomly enter chaperone-based refolding pathways or degradation pathways until the protein is repaired or destroyed. So here we have this example of E. coli folding a protein versus it degrading it. And so we call this a triage pathway because here we have, you know, DNA K binding to a protein. You have the ATP, you know, hydrolysis, and so it will essentially fold it. If it's still not properly folded, then it can be passed through a protease to degrade it. Or it can be fed to the GRO-EL for refolding, or it could just actually be folded properly and become a native protein. So there's this triage pathway. It can also be refolded via another chaperone. So this fold or destroy triage system is essential for a microbe because it allows it to survive these environmental changes and stressors. So there are some proteins that are destined for the membrane. So these are proteins that essentially require these special export systems or membrane transport systems. So I'm trying to think of if I want to kind of leave these next two slides for next week. Yeah, let's leave this, just these next two slides, because it's a little bit of a different, just a little bit different than what we're talking about. But now we're talking about actual trafficking. Remember, worksheet 3 is due on Monday.
I don't know. gym.
Anyways, I haven't done. I just don't know how to.
Can you email it?
I don't know how to get it. Like I don't know how to get it to PDF.
Well, you can email it in a Word document.
Oh, I can?
Save it, yeah, as a Word document, and then email it.
4:45 is like.
You can save it as a Word document, and then,
Okay. I had it done for like 2 weeks or something like that, or whenever it was due.
Yeah.
I just forgot to.
if you weren't here, then you have to send it to me.
Yeah. That's what I was trying to do. Forgetting it right.
Well, good luck on your exam now, so you wait till next year.
Yeah, I have it done. Yeah.
So have you saved it? Save it, file save. Yeah, so it's saved, okay, and then if you go in your email, no, so you go in your email and then you can attach it.
Okay.
Yep. Okay, and then, where did you save it?
Oh, you meant save the.
Worksheet 2.
Okay, perfect.
Yeah, perfect.
Thank you.
Yep.
So we made push deck.
Oh, cool.
I think it's just an algae. I do, but it'd be cool to look at. So
Yeah.
the roots that it was growing on.
Okay. Yeah, we can look at it. We can look at it if you want today after 2. That works. I'll be in the lab doing some other stuff.
Okay.
Or at 2. Yeah.
Can do that.
Yeah. Cool. I was gonna ask you. I was thinking about that. I'm like, should bring it?
It looks like it's just an algae, which is great, because
Yeah.
that's way better to have